Hi, I'm Jillian Cotter, a pediatric hospitalist at the University of Colorado. Today I'll be discussing our study titled Clinical Impact of a Diagnostic Gastrointestinal Panel in Children. Over the last decade, many hospitals have transitioned from conventional stool diagnostics to more rapid, expensive, and highly sensitive testing modalities, such as multiplex PCR gastrointestinal panels, or GIPs, that test for bacteria, parasites, and viruses simultaneously. The introduction of this new technology presents unique opportunities to improve pathogen detection and patient outcomes. It also creates potential challenges to diagnostic stewardship and high-value care if overuse of new diagnostics raises costs without impacting clinical care. Thus, it is critical to evaluate the utility of emerging technologies such as the GIP. In this study, we sought to evaluate the clinical impact of GIP testing by comparing test utilization, pathogen detection, length of stay, and resource utilization between the GIP and conventional diagnostic testing in a large pediatric population. We conducted a multi-center cross-sectional study of children who underwent stool testing from 2013 to 2017. We defined the GIP era as the 24 months after introduction of the GIP and compared outcomes to historic controls in the conventional diagnostic era, which included the 24 months prior. Both eras tested for bacteria, parasites, and viruses, but used different testing modalities. At our institution, the GIP tests for more than 20 different pathogens and replaced the need for many pathogen-specific tests that were used in the conventional diagnostic era. In this study, we compared test utilization, pathogen detection, and time to results between the GIP and conventional diagnostic eras. For hospitalized children, we compared outcomes including length of stay, use of ancillary abdominal imaging and laboratory testing, as well as hospital charges. We evaluated the percentage of patients who received antimicrobials for a bacterial or parasitic stool pathogen, excluding C. difficile, as this is discussed in a prior manuscript, and compared time to treatment between the two eras. We evaluated outcomes in the overall population as well as subgroups based on stool results and receipt of antimicrobials. There were 12,222 stool tests performed and 8,720 encounters. In the GIP era, there was a 21% increase in the proportion of children presenting for care who underwent stool testing. The GIP era had a statistically higher percentage of positive results, 40 versus 11%, and more positive results for each pathogen type, with viruses being the most common pathogen detected. In the GIP era, time to results decreased by over 24 hours, with a turnaround time of 4 versus 31 hours. Among 1,986 hospitalized patients, only 4% had a bacterial or parasitic pathogen, and only 3% received antimicrobials. For these patients, in the GIP era, time to treatment decreased by 25 hours, length of stay decreased by 2 days, and charges decreased by nearly $20,000 per patient. However, in the overall population and subgroups based on stool results, there was no difference in length of stay, ancillary testing, or hospital charges. Though this is one of the largest pediatric studies to evaluate the utility of the GIP, several limitations exist. First, our findings are limited to 4 hospitals within a single healthcare system, which may limit generalizability. Additionally, we use historic controls, and thus there may have been institutional changes between the eras that unknowingly influenced our outcomes. In summary, we found the introduction of the GIP led to increased pathogen detection and faster results. This translated into improved outcomes for only a small minority of patients, suggesting that unrestricted GIP testing leads to low-value care. Similar to other novel rapid diagnostic panels, there's a critical need for diagnostic stewardship to optimize the value of the GIP. Thank you, and please send me any questions or comments.